control FMD.
Okay, good afternoon, everybody. So here I'm going to give you a brief introduction about the current epidemiology of FMD and the control strategy in China. Three parts included in my talk. So I start with the brief introduction of where I'm currently working in. So this is the institute I'm working in, Lanzhou Veterinary Research Institute currently. So it is located in a geography center of China. So actually, the institute was founded in 1958. It was initially set up as a FMD research group, consists of around 10 people. Then later on, they grow up with different disease, different department. But the National Reference Laboratory for FMD was appointed by Ministry of Agriculture of China in 2002. And then it's in 2011 it was appointed as OIE reference laboratory for FMD. So currently there are about 61 staff, including 43 senior scientists in this institute, and there are 5 innovation research teams and a diagnostic group of 7 people under this umbrella of FMD reference laboratory for FMD. So the responsibility of these national reference laboratories as other reference laboratories in different countries. So we carry out the diagnostic service, including primary and confirmatory tests of diagnosis of the disease, carry out epidemiological studies, national surveillance, and characterization of the field isolates vaccination studies, and reporting to the authorities and the local laboratories, test development and the training, including national training and international training. So we have received trainees from Mongolia, Korea, Myanmar, and other countries around China. So we also organize a proficiency test scheme for the local laboratories at a professional level and even the county level. So provide consultancy to the authorities, to our government, and supply of reagents to different laboratories. So here I come to the epidemiology of the FMD in China. So the notified outbreaks since 2005 it was summarized in this graph. So here you can see from 2005 until 2009, there's H1 outbreaks. So the gray bar represents the H1 outbreak, and the red bar represents the type O outbreak, and the blue bar represents the type A outbreak. So from 2005 to 2009, there are H1 outbreaks, but no other serotypes except 2009. There's a co-outbreak of type A and H1. So from 2010 to 2014, there's type O outbreaks, occasionally mixed with type A outbreak. For the type A outbreaks was started reported since 2009, I mean recent years, 2009 to 2010. Then the rest have a quiet time for the next 2 years. Then this occurrence from 2013 until 2015. But 2016, this year, we only have the report of type O outbreaks. So this is the more details about the epidemiologic, outbreaks cases during 2015 and 2016. So for the 2015, they happened in 3 provinces, but they are all type A outbreak and belong to Southeast Asia 97 lineage or genotype. For the 2006, we have 2 outbreaks of type O. So one belonged to the Okasi top type, no one belong to Myanmar 98 lineage. So, so for genetic analysis, we can see for the type the type A, Southeast Asian 97 lineage, gradually since 2010, they evolved into the G2 class. Before then, it's a G1 class, so the virus is evolving and is drifting from the initial the virus. And for the type O field isolates, they all belong to Southeast Asian top type Myanmar 98 lineage, except on one occasion it was detected as O Cassie genotype or Cassie top type. For type O Pan-Asian has been reported during 2011-2013, 11 outbreaks in total, but no more cases since 2014 until now. So, as a reference laboratory, we also perform this kind of like evaluation of the vaccine, the current used vaccine, to see if we provide protection against the field isolates. So we perform this PD50 with the current available vaccine using the prevalent isolates GDMM 2013 as the challenge virus. So from the PD50 results, we can see all these currently available vaccine all above PD50-10. So it means it still works against the current prevalent circulating virus. So recently, I mean, early on this year, our Minister of Agriculture launched the National FMD Control Program for 2016 to 2020, those 5 years. And so the objective and expected results of this control program is about by 2020, the type H1 FMD should be free without vaccination across the country. The type A FMD should be free with vaccination. And the type O FMD free without vaccination in 3 regions, the Liaodong, Jiaodong Peninsula, Hainan Island. So these 3 regions, and free with vaccination in 6 provinces or cities, including Liaoning, but in Liaoning excluding Liaodong Peninsula, Heilongjiang Province, Beijing, Tianjin, Shanghai, and Jilin. So in these 6 provinces or cities should be free with vaccination. So this our goal by 2020 in that control program. So these slides show the animal service and the surveillance system in China. So under the Ministry of Agriculture Veterinary Bureau, there's the quite complicated system to form the good network across the country. So the green box shows the veterinary service and the detection and the diagnosis laboratory at a different level, at provincial level, county level, or even a town level and a village level. But the yellow box shows the National Animal Disease Surveillance station at the border area and the different places across the country. So from these slides, you can see, so the surveillance station or the animal service system in China is really formed into a very big, good network across the whole countries. In total, about 450 epidemic surveillance stations, formed this network across the whole countries and covered about more than 2,800 counties. So here comes to the summary of my talk. So far, there's no H1 outbreak occurred since 2010. And the current circulating strains of FMD For the type A, it belongs to Southeast Asia 97 lineage. For type O, it's Myanmar 98 lineage, and type O also has, type O is O Khasi top type and O Pan-Asian top type. But for O Pan-Asian, it's not occurred since 2014. So in general, the overall cases of outbreak are decreasing since 2013. So the current vaccine, including type O and A, is quite effective. So it still works across the country. The National FMD Control Program for next 5 years is in place. So I would like to thanks to my colleagues from the reference laboratory and the Ministry of Agriculture for the funding, and then CDC for the control of the disease, and the World of Reference Laboratory for FMD for help us to analyze the genome data, sequence data. So thank you.
Thank you for this very interesting presentation. Questions? Yes, here. It's a big room.
Thank you. To be honest, this is the first time I'm listening something from China, and it's interesting for me. Why your calendar starts from 2005? What happened before that? Is there any information? This is just if you know. And is this only one, I mean, how many production units you have? Is it only one institute? Can it satisfy all the needs for this huge country? I don't know how you are going to organize this if
So the first question, yes, there was the outbreak of FMD before 2005, but it's not reported cases. There's no systemic reported cases can be calculated or submitted. No submitted cases. I think before then wasn't OIE reference laboratories, so the data wasn't collected. So. That's why we couldn't have this data presented, Your second question is, what's the population size? Oh, sorry.
How many production institute for production of vaccine you have? Could they satisfy all the needs of the country? Because you have ambition program and
Yeah, we have the compulsory vaccination program, the vaccination campaign across the country. So about 4 billion doses per year has been produced, every year, annually. That's for vaccine. Yeah, 4 billion doses. Yes, a lot. So yes. We have a large population of domestic animals.
It's another scale. Chris?
Yes, if I may, we have seen some presentations in this session on Saudi Arabia, where it was clear that not always infection is reported. Georgia has also indicated that although no clinical disease has been seen for a number of years, NSP antibody levels are relatively present. Earlier this afternoon, we saw information from OIE about cattle movements going across Southeast Asia into China. So can you also share with us maybe your serology data, or are you doing any serological surveillance?
Yes. So we have so many surveillance stations. So their work is to do the post-vaccine monitoring surveillance, as well as the active surveillance. But I didn't got data here because these years we haven't completed. And the last years, they said it's published in our website from the Bureau, Veterinary Bureau. So in general, they said it's about 80% of the population has above the criteria. So it's quite good coverage.
Okay. Quick comment, maybe?
Well, maybe I haven't been clear, but I'm looking not so much for post-vaccination monitoring surveillance, but more like what is your non-structural protein levels of, as an indication of virus circulation. So an understanding of what FMD virus circulation is there, although you may not have clinical reporting.
But you know, for during this surveillance, as I said, it's not only passive; it's also active surveillance. So we as a reference laboratory, we random sampling the serum and probe samples from the slaughterhouse. Occasionally, we found some PCR positive, but hardly, got to the other correlated data to support if this is positive or not. But in terms of NSP antibodies, we do test against the serum. Yes, some occasionally, again, there's some positive, but it cannot really form a whole story. But at a provisional level, they also doing the NSP antibodies during the surveillance. If they found antibody positive against the non-structural protein, they will go to the farm to check the clinical signs. So they follow the procedure to do for the investigation.
Thank you. A very quick last question.
I just, sorry to interrupt. I just want to add something further to Dr. Lee's answer. It's about the question about the cattle movement from Southeast Asia to China. Actually, the main poultry for those cattle entering China is Yunnan province. And in Yunnan province, there is a very strong surveillance there, just on the border area, especially focused on the main cattle markets and the slaughterhouses. So the researchers there, they perform routine surveillance there. They collect the lymph nodes from slaughtered cattle, and also they perform serological tests. And and I think in one of the OIE's meeting, they presented some data because those are their data, so I don't have the right to present them in my presentation. But according to my recollection, I remember in their serosurveillance, they say for those animals that are originating from Southeast Asia, it seems like the positivity rates to the 3 serotypes ranging from 10% to 30%. So, but this is not very sure. Because they detected the antibodies to the 3 serotypes in ALP, Lassa, and also they performed NSP. But they said some animals in Southeast Asia may have previously vaccinated. So those seropositives does not necessarily mean those animals are affected by FMD. Probably they have been vaccinated several times previously, and it's just introduce some positive antibodies in the serum. And about the pathological examination, I think the positive rate is relatively low, but of course it depends on the sample size.
Thank you. Thank you. Thank you, Yamini. I think we need to move on. Time is going, and we pass to the next presentation. But thank you.